So it asks you to label the ORIC, which is the origin of replication in bacterial cells. So that's right here. And then the DNA primase is going to set down the RNA primers that has the 5 RNA bases. So here's the RNA primer here, and primase is right here setting it down. And then you have helicase, which is going to go right here. And that's going to be unwinding the 2 strands of DNA so that replication can occur and that can have enough room for that to occur. And then, like we were saying earlier, the RNA primers are right here. So there's one right here, down here on this Okazaki fragment, over here. And so these RNA primers are necessary for that replication to begin. And here you have the leading strand, and the way that you can tell that it's the leading is it doesn't have those fragments like down here, these Okazaki fragments. Leading strands just have that continuous strand, and so it's a little smoother. And then the lagging strands have those Okazaki fragments. So the DNA polymerase 3 alpha subunit, which is right here, he's going to go down these strands, so there's another one right here, and synthesize the new DNA strand. And he's going to do the same thing over here on the Okazaki fragments. And then what DNA polymerase 1 is going to do is he's going to come and take these RNA primers off and replace it with DNA, so there's no RNA in the newly synthesized strands. And then with the last part, it asks what the Okazaki fragments are, and those are the segments of DNA on the lagging strand that are being synthesized.
Beauty. Okay, let's do number 2.
So number 2 is one of the fill in the blank, and a lot of this comes directly from the reading packet. But with number 1, DNA primase, its main role is a protein complex that synthesizes primers in eukaryotes. And a key way to remember this, if you just remember the A's at the end of the word means to synthesize, and then DNA primase, so it synthesizes primers. That's one way you can remember that. And then the DNA A proteins, its roles is separate the AT-rich sequences within ORC. You get that from the reading. The epicene subunits is the subunit of DNA polymerase 3 that contains proofreading activity. And then DNA polymerase
Epsilon.
Yeah, that's the same thing, epsilon. This protein synthesizes the leading strand in eukaryotes. And then the TUS, that's the protein that displaces DNA polymerase 3 from the chromosome to terminate replication. DNA polymerase 1, if you remember from up in number 1 with the drawings, its role is to remove the DNA, and it does that in it removes it in the 5 to 3' direction as well as synthesizes in the 5 to 3' direction. And then topoisomerase type 2, this will travel in front of helicase as it unwinds and unwind the DNA, or also called negative supercoiling.
So it doesn't get too tightly wound in front of it.
Yeah, exactly. And then ORC, this protein binds directly to the ARS element. We learned that from the reading. And then the SSBPs, these proteins prevent DNA strands from annealing during the replication.
So it's the single-stranded binding proteins.
Yeah, they just prevent the DNA from recollapsing and rebinding on itself as the helicase goes and unwinds it. And then ligase, this enzyme seals the Okazaki fragments into continuous strands of DNA. So it goes after DNA polymerase 1, goes and replaces all the RNA with DNA. Ligase will go and seal the strand, so it's the last step.
So you can look up here, and the Okazaki fragments will look like the leading strand after ligase has gone through.
Beauty. So we can probably just highlight these ones.
Sure.
So we know from the readings there's not a lot of explanation to go into this, but the enzymes that consume ATP during replication are DNA helicase, gyrase, primase, ligase, and MCN. The 5 enzymes that consume DATP during DNA replication is the DNA polymerase 3 alpha subunit, beta subunit, epsilon subunit, gamma subunit, and the DNA polymerase 1. The 5 enzymes that have a 3 to 5 prime exonuclease activity is DNA polymerase 3, 1, the epsilon subunit of DNA polymerase, and the delta subunit. Part D, the enzyme that's involved in replication that uses DNA as a template, so it will use the sequence from DNA to synthesize RNA. So it's not going to be any of these, it's going to be DNA primase. And then the enzyme that uses RNA, so this one uses DNA. The one that uses RNA as a template is telomerase. Yep, we are. So.
Okay. So with this one, we're just going to be synthesizing DNA, and it's going to give you specific DNTPs that you have to use. So basically, the easiest way to remember this is that if it doesn't have the structure of where it's D and then some base TP, it won't be able to be used to synthesize DNA. And so first, it's asking us to not include the primer. Yeah, so it's asking us to not include the primer. So we're going to figure out where this primer is located in this DNA sequence up here. And so if you look at it, we're going to have to switch it the other way because they're both going from 5' to 3'. And so we know the complementary is going to be opposite. And so if you look here, the easiest way to find it is you see these 3 A's, and then you see these 3 T's up here. And so you're able to kind of distinguish that this primer is going to fit right here. And you can kind of see that those complementary base pairs. So we know that we need to start synthesizing DNA right on this T base. And so here we've been given all of the complementary bases that we need. So we have A, C, G, and T. And so we know that the whole complementary strand is going to be created starting at this T base. And so we're going to get, going from 5' to 3', we get A, C, T, G, A, T, GGT, CCT, GC, TAT. And so what's different about part B is that we have DCMP. So any base that has the complementary base of this C is not going to be able to be translated. So we look up here and we have a T first, so we have the A down here, but then our next base is going to be, the complementary base is going to be a C, which can't be synthesized because we don't have the DCTP. So that is only going to synthesize a strand with that single base A. And then with this one, it looks like we do not have the base necessary to synthesize the T. So when we get to this A base here, we're going to run into that problem, and that's why this one is only going to be 2 base pairs long. And then finally, with this one, it says GTP, which isn't even going to be able to be used as far as synthesizing that G nucleotide. And then we're fine here with the DTTP. And so we can look, and it looks like we run into that C base on the 3rd nucleotide there, or on the 3rd base there. And so we know that we're only going to be able to transcribe the T, C, and the A, which is going to give us the complementary strand of ACT.
Okay, so for number 5, we found the easiest way to do this one is after you go through the reading, just if you can remember or go back through and just write next to each one of these which one is involved in prokaryotes, so which one is involved in eukaryotes. And so from the reading, you can go through DNA gyrase, that's found in prokaryotes. The TUS, that's a termination factor in prokaryotes. DNA A, that's found in prokaryotes. DNA polymerase epsilon, that's found in prokaryotes from the reading. DNA primase is found in prokaryotes. DNA polymerase 1 is found in prokaryotic cells, as well as DNA polymerase alpha subunit. The triple DNA polymerase 3 alpha subunit, that's found in eukaryotic cells. And so those are prokaryotic proteins.
Okay, so for the second one, it's the same principle. You just have to go through the reading and find the ones that are found in this instance, we're looking at the mouse mus musculus. So the ones that are covered in the proteins that are found in there are the MCM helicase, the CDC6, the DNA A, the DNA polymerase epsilon unit, the DNA primase, and the polymerase alpha subunit, and pCNA and telomerase. These other ones, the DNA polymerase 3 alpha subunit, DNA polymerase 1, TUS, and gyrase are not going to be found in the mouse. They're not proteins that are found in the mouse mus musculus.
For number C, it asks which of the above proteins is involved in chromosome compaction, and that's the function of DNA gyrase. That's its function, is compaction.
Beautiful. And the proteins that are involved in initiation in DNA replication are DNA A and CDC6.
And which one of these proteins is involved in the termination stage? Remember, that's the function of the TUS and the MCM helicase.